Hi everyone, I am Xinran Xu, a PhD student work under Dr. Abhinav Mishra from University of Georgia. My topic today is the association between pastured poultry-related microbiomes and Campylobacter presence. Microbiome is a complex and dynamic bacterial community. These communities interact with host, other animals, and the environment. For poultry production, environmental factors such as hatchery hygiene levels, housing, or litter quality and management can significantly influence poultry microbiome. Studies have shown that foodborne pathogens can colonize chickens at any stage from farm to fork. Also, the source of pathogen is not only from farm environment but also from other chickens, like within a flock. While the conventional U.S. poultry production continues to grow, there is an increasing demand for alternatives such as pastured products. While there is limited research available discussing pasture microbiomes and the resulting foodborne pathogens within this production system, as a result, it's important to understand the dynamics of pasture poultry microbiomes and its connection to foodborne pathogens. Feces and soil samples were collected from 11 pasture poultry farms from southeastern United States from 2014 to 2017. Microbiomes of these samples were characterized using Illumina 16S rRNA gene applicon sequencing. Meanwhile, Campylobacter of these samples were tested for presence and absence by cultural methods. Feces and soil samples were taken 3 times, start, middle, and end, throughout a flock's life cycle. 2 types of models were developed by pasture times separately for feces and soil samples. Linear discriminant analysis effect size was performed to select the differential features between Campylobacter present and absent groups. Random forest was conducted to predict Campylobacter presence and absence by microbiome taxa features. Figure 1 shows the linear discriminant analysis effect size plot of feces samples, where A is at beginning and B is middle and C is end of the pasture. Formicutes can differentiate Campylobacter negative group from positive group at the beginning and middle of the pasture. In another study, they investigated Campylobacter jejuni colonization using Zika samples. They found higher amounts of formicutes, where the average was 62% in Campylobacter negative samples compared to Campylobacter positive samples, which the average was 63.6%. The results agrees with our data. Figure 2 displays the top 10 taxa selected by random forest that can best predict Campylobacter presence and absence in soil samples at the beginning of pasture time. Microbacterium species was identified as the top taxa that can best predict Campylobacter. These species have been described as the free-living opportunistic alpha proteobacteria. Microbacterium species are numbers of the soil microbiome. There are no results discussing the link between Microbacterium and Campylobacter, but our results indicate that these bacteria could be a good indicator of identifying Campylobacter in soil samples in pasture poultry farms. In conclusion, these models provide an alternative way of identifying and differentiating and predicting Campylobacter presence in pasture poultry farms. The results show the potential of integrating microbial community data in predictive microbiology and in risk assessment models. Thank you so much for listening.